minutes here, and I don't know why that's not scrolling. Then on March 28th, we'll have Glenn Branch from the National Center for Science Education talking about a child's garden of climate change denial. And on April 11th, we will have Warren Ullman, the director of the Paleontological Research Institution, talking about his recent trip to Antarctica. It's actually got a different name than that I need to update in the slides. Our Earth Day program will be on whales in protecting whales from getting caught in fishing gear for April 25th. And the National Earth Day theme this year is plastics in the planet. So we'll be talking about that, and that full description of that should be up on the Science in the Virtual Pub webpage in a couple of days. Then we'll round out the spring season with talking about the JOIDES resolution with a panel of educators who have sailed on the JR. And I think I'm going to go ahead and turn it over to Toby. You ready, Toby? All right, I'm going to stop sharing my.
All right.
Take it away, Toby.
Thank you. Yeah, sorry, I could not get logged in. And I've tried to set this up. So we're going to do something new here that I've never tried before because this is science. So in a second, I'm going to turn everyone over to into breakout rooms. Before I do, I want to just get a sense of, we've got 15 people here. And in the chat, can everybody use the chat?
Yep.
Great. So we'll do a little bit of science before we get started. We'll do science and then we'll do some more science. So I'm a professor at Cornell in the Earth and Atmospheric Science Department. I teach a big climate and energy class in the fall semester, and I've been working closely with Rob and Don and Ingrid and Alex for the last, really since the pandemic, and we started collaborating pretty intensively on some of these projects. And one way that we've been using my large class in the fall is a bit of a lab, a test case for climate science themes that land well with young people. And so this is back in the days of the pandemic. We were all online. And one of the projects that landed well with young people was a project that, like, my children and one of their friends had invented, which was to do a CO2 drawdown experiment inside of an aquarium. And now everybody, I think, who has taught or thought at all about photosynthesis may expect this to be a relatively easy challenge. And this is the challenge I'm throwing out to you all today. And it turns out to be not an easy problem at all. And over the years, it's been kind of fun because I've had a handful of successful tanks where I was able to get CO2 levels to go down. And I brought a bunch of material in with me today. And we think this is an activity that works pretty well in the classroom. We've done it two different ways. We're going to do it the way where we try to draw CO2 down inside the tank. The other way that we've done it recently is to light a candle inside the tank. I'll talk about that in a little bit. But what I have is a bunch of plants and mosses and algae that I've arranged in my lab. And the reason I was running late is because I was trying to set up a webcam so that I could show you everything. And I'm going to keep working on that when we do the breakout rooms so that I can actually show everybody around. But so our science experiment that we're going to do, that you can hopefully try to do some version of in your classrooms, is going to start with a prediction. And the prediction I want you to make, you can just do it in the chat right now, is about how high do you think CO2 levels are going to be inside. Here, I can turn the camera. This is the tank that I've got set up for us. You can see there's some tropical plants in there. There's a potted plant. There's some mosses. I'm going to open it up, put in stuff that at your request. I'm trying to use this as if you were here in person. And if you were here in person, I'd let everybody choose something to put in the tank. The first prediction I want you to put in the chat is, how high do you think CO2 levels are before we even start this experiment, right here in my office? So in the chat, how high do you think CO2 levels are right now? And then I'll put the sensor in there and we can get an answer. First guess is 500. I'm the only human in the office. I'm full of hot air sometimes. I've been talking for a while. $450. Two guesses. Get some more. Let's get at least 3 or 4. The plants have been in there for about a week, almost a week. 475. It's a little bit higher than the environmental background. Different units. Fun. Most of these units, I assume, are in ppm. All right. 350. Well, we're about to get data. I'll show it to you as soon as it comes online. It's in there now.
There's a question if you've got an open window in the office or not.
Nope, no open windows. All right. Well, let's find out. I just put it in there. It takes about a minute or two to connect. And while it's doing that, I will set up the breakout rooms. And in this case, what I'm going to do is I'm going to connect it to the Wi-Fi on my cell phone because we've had a lot of success getting that to work in classrooms so far. That's usually the easiest way to get some of these devices to connect is just set up a little hotspot on your phone. So that's what I'm doing now. You can get data. And in about, I think, less than a minute, we should have some. Yeah. Suspense. This is where we need some sort of dramatic music playing. So cue up the Avengers score. All right, 240 is the correct answer. 240. So it was a very successful tank experiment, actually. One of the more successful ones. Now, it hasn't been continuous. It was probably higher earlier in the day today. I didn't tell everybody this. I could have revealed this information sooner, I suppose, rather than right now. But I actually have a westward-facing office. And so for the last 4 or 5 hours, there's been direct sunlight pouring in on the tank, which helps explain those pretty low numbers. And the experiment we're going to run now, if I can get my webcam to work, show you what I've got here. You all can choose. I'm going to do, we have 16 people, so I'll do 5 breakout rooms with about 3 people in each breakout room. And webcam now works. There we go. Yes, it's working. Okay. So there's my westward facing window. And then I've got loads of different mosses. Those are all mosses that I've collected or students have collected for me. Got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 clumps of mosses. We've got a tropical plant in there, a palm. We've got one of these that probably have seen, a common house plant. We've got some more moss that's already in the tank. I have this moss that I've been jokingly calling the Rob Ross moss because we collected it from the Museum of the Earth in the fall and we've kept it alive all winter. Been growing vigorously. And then over here, I have a nice tank of algae that I can add some of this to the tank to see if that helps our experiment. I also have a larger giant bottle of algae. It's like probably at least 2 or 3 gallons of water with algae, also collected from the Museum of the Earth. And then I have some aquatic plants. I have some air plants. And I have some sand, just sand. So in your breakout rooms, I'm going to set these up, and I want everyone to decide as a breakout room what you want me to put into the tank. So spend about, I don't know, 5 or so minutes on discussing what of everything that I just showed you, and I'll put that all in the chat right now. You can determine how many clumps of moss. So up, there's 12 clumps plus another. I think there's a clump of moss. I tried to have 15 clumps total. I think there are actually 16 clumps of moss. So everybody can put their own moss in it. And the idea is that I'm trying to represent this in the way that you, as a teacher, would be able to have students, every student in the classroom or groups of students in the classroom, collect something, bring it in the tank, and contribute. And I did this exact experiment with my son's classroom when he was in 5th grade last year. I had everybody in his 22, 23 students in the classroom. Every single one of them collected something in a little jar, brought it into the tank, and then we looked at what happened to the CO2 levels over the course of a few weeks. Some unexpected things happened because those weeks coincided with historically poor air quality events in the Northeast last year. I'll talk more about that and show the data from those experiments after the breakout. So imagine this is, so if you're teachers, you're sending your students out to go collect the items. Well, we're online, we're doing this virtually. You're going to decide what items that I've collected are going into this tank. So I'll list it here: moss. There's a palm tree, little mini palm, house plants, algae, air plants. I'm going to write pond, PRI pond algae. 2 different types of algae. And aquatic grass from the PetSmart. All right. So now the breakout room should be ready. And I will see you all again in about 7 minutes.
Say again what our instructions are in the breakout room.
You are going to decide what you want me to put in the tank before we run our experiment. Then we're going to run the same experiment that I just ran this afternoon. So I got down to 240. That's the benchmark. So this afternoon, I set up the tank with a bunch of stuff in it, a bunch of moss, some tropical plants, house plant, the Rob Ross moss, and I just put it by the window in my office, not an open window, sealed it up, and we got down to 240, which is pretty good. So can you beat my score? No, you won't be able, I don't think you'll be able to beat my score in a short period of time, but hey, stranger things have happened. And it looks like our sunlight might be going away. So I also have a grow light that I'll set up so we can be sure to get a nice photosynthesis response. All right, ready, set, go.
Hi, Rob. Are you back?
All right, everybody's back? Looks like everyone's back. Okay, so now we've got a Google Doc here. Hopefully everyone noted their room number. If not, no big deal. Just go ahead and you can maybe coordinate this in the chat if you don't remember your room number. I should have told you to remember your room number. Go ahead and list the items that each room has decided should go into this tank in that document so I can use it. And then we'll come back to it and I'll show you sort of the scope of the project and where things stand and some other resources that PRI has put together for this project. All right, it looks like this is being populated. Perfect. Awesome. And like I said, this is an experiment inside. We're going to do an experiment, but this is also an experiment. How well can we do this online in real time? Never been done before. Y'all are part of history. And if we, if we save the world with this approach, you will have been there on the ground floor. All right, houseplants and moss. Algae, moss, air plants. All right, so I'll start queuing up some algae here. All right, more everything but the eggplants. Oh, so this is what happens. This is perfect. This is exactly what happens when we get to the students. It's great. So we have a debate about what should go in. Let's keep it going. Yes, our rooms numbers taken. We limit our plan. Okay. All right. So it looks like there's like every these 4 mosses. I'll compromise and put in 1 houseplant. And then the fun thing that I've planned for today that I've never done before is I'm going to put the camera, and we have 1 camera inside the tank. So we have like a tank cam. And then we want the houseplants. We want to find a way to compare the carbon collection moss to algae in the plant. Yes, that's where we're headed to. We won't be able to figure all that out tonight, but that's the goal. It looks like one more thing of algae. Little cute little bamboo if you had it. I don't have bamboo, but I'll put in the aquatic grass in its place. All right. I think I've got that right. So now you all are going into the tank. So who here Who remembers the Biosphere 2 experiment from the early 1990s? Anyone? Show of hands. See if I can get this lined up correctly. There we go. A few people. All right, you're all biospherians now. This is like Biosphere, I don't know, N. Now you're in the tank. That's my bike outside. There's the air plant. There's moss all on the base of the tank. And there's the algae and the aquatic plant as the stand-in for the bamboo. And now, the air quality monitor is going to go in there, hopefully where you can see it, and not blocking any of the... And if you can't see the display, that's okay. We have a workaround for that. Because all this data is going up to the cloud in real time. There you go, there's the display. Not close enough that you can read it sometimes? You read that, Don?
Almost.
Almost.
Yeah, I can read it if I try. And
I mean, I'll pull up the web page for it as well. CO2.
Yeah.
So it's
and folks should go to speaker view if they're not on there already to make bigger
right.
Toby's camera bigger.
So CO2 is already at 6.2 probably from me breathing. So let me air out this thing before we seal it up. And we will use the grow light since the natural light has ceased.
62.
All right.
And I'll note while Toby's doing stuff that I recognize the sound of the cover he's using. It's an aluminum cover from the catering from the Saturday's workshop.
Yeah, so this Saturday, what we did was we lit a candle inside the tank to see how long we get to burn for. Because the more photosynthesis is happening, the more oxygen, the longer the candle burns. So without any plant material inside the tank, the candle burned for about 5 minutes. And on Saturday, with plant material in the tank, it burned for 17 minutes. So a huge difference. And so if you don't have a CO2 monitor, you know this is too tedious to try to set up, that's a nice one to show the effects of plants on the atmosphere. All right, so now we're going to get the grow light up and running. And then I'll show you some data in nearly real time, as well as the PRI page for all this stuff. Okay. All right, I'll switch you back over to tank view mode. So you can see that there really now is, so this is the grow light being off, the grow light coming back on. We'll check in with the data that we get. So in the meantime, while we're waiting for that, we have put together a web page. I'll send that out for everybody. I'll do another set of breakout rooms here. Should be able to see that link in the chat now. Okay, you can see it?
Yep. Yeah, and I actually put it in there a few minutes ago. So
great. Okay. So check out that web page. Now we're going to do breakout rooms again. And this time, I'm just curious to hear your perspectives. You look through the web page. How might you use or modify this type of approach in teaching, and also at what level? And so we'll do a discussion of all that when we come back in just a few more minutes. And I'll look at the data. That'll give me enough. That'll give us 5 minutes of data to see what the trend looks like from this experiment. And if we're lucky, the fact that there's intermittent sunlight will show up in our data. That would be really cool. Doesn't always happen, but I set one of these up in my garage, and there's a very clear signal. When sunlight was shining in the tank, CO2 levels were going down. And when the sunlight went away, like either because the windows were at a different angle or the clouds went past, CO2 levels started going back up because of respiration. All right, ready, set, go. All right, looks like everybody's back. So let's look at our results so far. I guess before we do, who thinks they're going to have gone up? Oh, show of hands for if they've gone up. Thinks they've gone down. All right, let's find out. So data has been uploaded to the cloud, and I just created, while we were in the breakout room, what's called a lab. I set the time bounds to be from about 5:30, so 12 minutes ago, and this lab experiment will continue for another 12 minutes from now. And they've gone way up. They've gone way up. And this is not an uncommon pattern. In fact, this is exactly what happened inside Biosphere 2, the experiment that I was talking about in the 1990s. They had envisioned building essentially a giant greenhouse in the desert that would be a test bed for technology and ideas to terraform or inhabit other planets like Mars. And so it was supposed to simulate, the Biosphere experiment was supposed to simulate the challenge of living on a foreign planet in a closed environment, in a closed system. And so they brought in all kinds of tropical plants and agricultural plants and a bunch of topsoil, and had, I think, 12 or 13 biospherians close themselves inside and try to survive off of food that they could grow inside Biosphere 2 and harvest and raise and slaughter animals. And it did not go very well very quickly. And the reason was because they brought in so much topsoil, the soil microbes started chewing up all that yummy carbon, yum, 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 and releasing CO2. So the soil respiration rates inside this tropical environment that they created vastly exceeded the photosynthesis rates. So the net primary production was, in effect, negative. And if you've ever been to the tropics, there's just not a lot of topsoil. There's not a lot of that black, thick topsoil, sometimes called humus, in this part of the world. You just don't see that in the tropics. And since they had a tropical environment, lots of humidity, lots of warmth, microbes were very happy to just chew away at all that carbon. So CO2 levels started to go up above 1,000, 2,000, a few thousand, and then they had to end the experiment because oxygen levels were decreasing from 24% normally to like below 20% to closer, I think, to at the end of 10% before the biospherians started to panic and they started injecting oxygen. But it's still an amazing facility. You can go visit it today. The University of Arizona has taken it over. And I think there's some lessons here for what happened today. So I've done this. Here, let me show you so you don't feel bad. Don't feel bad at all if you feel like this is an unsuccessful experiment. No such thing as an unsuccessful experiment unless you fail to learn something. So here are my favorite experiments. There's about, I think, 30 some of my favorite ones. All of the experiments I've run, total over 50, I've only successfully gotten CO2 levels to go down inside this tank less than a third of the times overall. But what I learned by doing them with my kids and with various classrooms, including this classroom that I told you about last summer, is that as soon as you start putting soil, I can point to the culprit here. I know who the culprit is. As soon as you put something that has soil into these experiments, the respiration rates exceed the photosynthesis rates. And so it was probably the houseplant, which is potted. It's relatively fresh soil, which means there's plenty of oxygen, and all those microbes are doing the same thing they did in Biosphere, just yum, 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 chewing up all the carbon, releasing it into the atmosphere. And when we did this at the museum, we did it with, we just let people come by, we had a little station set up. We let people come by, visitors to the museum, design their own little worlds, and walk around the museum for 15, 20 minutes, come back and see how things did. Some were successful. The ones, there was a couple that came by who were aquarium enthusiasts. They kept tropical fish. And they had an intuition that aquatic plants, algae, moss with almost no soil would be the best way. So that one was successful. CO2 levels went down in their tank. Other ones, people thought, oh, tropical plants like a palm tree, a little miniature palm tree, or the large leaf area of some of these plants, that should definitely do a lot of photosynthesis. And while it's true, it does do a lot of photosynthesis, the respiration rates tend to outpace the photosynthesis rates. So now, if this were a classroom, and we just done this experiment, and this is kind of what we did with my son's classroom, is they went up to 3,000 ppm, so don't feel bad. You did better than they did. What we would say is, okay, well, let's figure out, let's actually start doing the science now. So now we have a result that tells us that as a whole, this ecosystem that we created, this little miniature world that we've created, is on average seeing a net increase in CO2. But could we go through systematically, week by week, and isolate which components are effectively doing photosynthesis and which ones are causing respiration rates to go up? And you could even have just like straight up soil. Just put soil, moist soil into the tank and see what CO2 levels do. We did this with leaves on my own. We did this with soil. And in both of those cases, the respiration rates were about the same as when we did it with just a ball of pizza dough. So the yeast in the pizza dough is releasing CO2 at about the same rate as the soils in our experiment, which kind of makes sense, I guess. And also, don't feel bad. Like, so here's try to find one where I used what a Cornell scientist told me I should use. Plantopia. Yeah, I think this is the one. No, this isn't the one. This one actually worked kind of well. All right. CO2 levels went up. We had this huge diurnal cycle. This is the temperature at the top here, humidity levels, and then CO2 levels started, they got up to like 2,000, and then they started to come down with a huge diurnal cycle. Let me find the one where solicited feedback from Cornell scientists, and it's not showing up now. So I'll just jump straight to the results from my son's classroom last year. In any case, they told me, there's a Cornell scientist told me, oh, they're like a biogeochemist. They said, well, you should put grass, because grass in the springtime grows like crazy and definitely will draw down CO2. And I did, and CO2 levels went up. And I told them 3 weeks later, CO2 levels like reached 10,000 ppm. What's going on with that? They said, oh, that doesn't make any sense. You just grass from your yard? And I was like, yeah, it's just grass from my yard. I didn't do anything. I just dug it up and put it in there. Like, oh, because you dug it up, you exposed all those microbes to oxygen. CO2 level from the microbes in the soil would account for the CO2 levels increasing. So here's the one that we did in my son's classroom. And we had two eggs right next to each other. So one was inside the tank, and the other one was outside the tank. And the one inside the tank got to about 3,000 before it started to come back down. The one outside the tank has this diurnal cycle where CO2 levels go up during the day because there's kids in the classroom. So the control tells you about people being in the classroom, and the test egg that was inside the tank tells you about what's actually happening in the tank. So it went up to about 4,000, then started to come back down. So sort of like a Jurassic World. But the really interesting thing that happened was that inside the classroom, and because of the smoke event, suddenly the egg that was inside the enclosed tank became like the control, and the tank that was sitting out in the classroom recording air quality became the interesting thing to watch. And as the smoke moved in, air quality values across Ithaca went above 200, just unhealthy. And you see that reflected in these sensors, which just happened to have been placed ahead of time in the classroom. It also tells you that the filtration system, which had been installed for COVID, might be good for keeping air circulation healthy by bringing in outdoor air, but because it was bringing in outdoor air, it was also increasing the air quality, worsening air quality and increasing the particulate matter, fine particulate matter inside the classroom. All right, all right, so we got like 5-10 minutes left. So I wanted, I guess, I don't know if people can put in the chat, or if you want to just have a conversation now, we can raise hands or just talk. From what you've seen so far, either from the page or just from this little short demo, which, you know, like I said, it was a real experiment. I didn't rig it ahead of time. I tried to be as realistic as I could with what you might encounter if you do this in your classroom. It's not guaranteed to work every time. I can tell you that having soils in there generally makes it not work. If you can figure out a tank, if you can design an ecosystem with your students that has some soil and pulls CO2 levels down, that would be amazing. You'd be the first. You'd be the first one who pulls it off. In principle, it should work if you keep the tank really cold, because in high latitudes, soils can be, operative word is can be, net sinks for carbon dioxide. Plants grow, they die, they start to decompose, soil builds up, that's pulling CO2 out of the atmosphere and putting it in soils. So far, with our experiments, the presence of soil virtually guarantees that CO2 levels are going to go above a few thousand ppm, and your world's going to look more like the Jurassic or the Cretaceous or something, and not successfully pull CO2 levels down. So either, yeah, so we got 5 minutes. Let's chat. Let's talk. What do y'all think?
Can you talk about the candle experiment?
Sure, I can actually show the data from the candle experiment, I think. But what we did for the candle experiment was, if I had a match, I could actually do it now. I have a candle, but I don't have a match. For the candle experiment, what we did was same basic setup, but we lit a candle, let it burn inside an empty tank, and it burned for 5 minutes. And then we invited the teachers over the weekend, and I also did this with a class of 7th graders. I don't know if she's here. Is Michelle Welsh here? Can't tell. Her class of 7th graders, we did this 5 times in a day, where she had, I think, about 18 tables, 2 kids per table, 16 tables, 2 kids per table usually. And the same sort of setup that you did with your breakout rooms, we asked them to discuss amongst themselves what they wanted to go grab, and then sent 1 kid from each table to go pick out moss or algae. I only gave them the moss and the algae options because I wanted to like rig it so they would work. And they brought it back, and then they put everything in the tank, piled up moss and algae as best we could, sealed it up with a piece of metal, aluminum foil, because with plastic, the candle will melt the plastic, and if you flip the tank upside down, I've had this happen, it will break the glass in the tank. It's the heating, so you have to use aluminum foil. Sealed it up. I told some silly stories about my first science experiments as a 7th grader trying to build a rocket car. And we let the candle burn. Everyone's like very intensely focused on the candle burning. And it burns for a long time, burned for almost 10 minutes. So twice as long in her classroom as the control. And they understood the concept of a control. And they understood that the candle flame went out because it ran out of oxygen. And they understood that because there were plants doing photosynthesis, releasing oxygen, the candle burned for longer with this experiment. So now she has 2 of these sensors in her classroom. She has a bunch of material that I left, and they've also got a pond. What they're going to try to do is a long-term, like a week-long or 2-week-long experiment with each of her sections to pull CO2 levels down by setting up an experiment like this and letting it run for a week or 2. The question is the plan to use hydroponics to grow food on Moon, Mars then, so they don't have to use soil? I mean, that seems like a one take-home message that you could learn from this experiment. I think another one is one of the questions I got this is from a high school student, clever high school student, said, well, isn't there like he didn't say it quite like this, but he asked, isn't there a negative feedback when we think about climate change? I talked about climate change and the risks of climate change and how these experiments that we're doing now are really geared towards thinking about the second half of the century, when we're going to have to find a way to get CO2 out of the atmosphere if we want to fully stabilize the climate system. And he said, well, what's the big deal? I mean, isn't it just going to mean that we have longer growing seasons and plants are going to do more photosynthesis? And after having run these experiments, I said, yes, that's probably true. You'll have longer growing seasons, more opportunity for plants to grow. However, you also have longer warm seasons for soils to do respiration. And so those longer growing seasons aren't necessarily going to be fully advantageous if soil respiration rates mean that you can also release more CO2 by having more soil be metabolized by microbes. All right. Some more questions have come in. There's talk about not having exposed soil with the plant so that it can sequester carbon. So, oh, sweet. This is a great idea. Cindy has an awesome idea. And I think try it out. We don't know. I mean, we have only done a very limited, narrow scope of what's possible. The next question is the sensor. So there's a few options for sensors. You can buy them online for as little as what, like 20, 30 bucks. If you just want to do if you just want to have a sensor that doesn't connect to Wi-Fi and CO2 only, they're not very expensive. The ones that we use are quite a bit pricier. They're from this company called Air Quality Egg. We're working on a proposal and a mechanism where we can lease them out from the company and then loan them out to schools. We also have a handful of loaners. Most of them are already spoken for the rest of this year, but there should be some available ones that we can loan out next year. We might have a handful towards the end of this year. Yes, drought. So actually, one of my main areas of research is drought. So one of the other interesting things that I learned from the person who donated the air plants is that they actually don't open their pores until night. So they will do photosynthesis in the day, but they keep their stomata closed. And then it's not until night that they do the gas exchange. So it'd be interesting to do a more tropical experiment with just like only air plants and see what happens when the lights are out. And yeah, drought is a water stress. We have, I didn't set this up for today, but we also have soil moisture sensors that we're putting up at the Museum of the Earth and at the Nature Center. And we hope that in the not so distant future, some of you and some teachers across the region will volunteer to have soil moisture sensors at your schools that will in fact help us do drought forecasting.
In Arizona, in the desert, they have at night, we have creeks that run that are dry during the day because they open their stomata to,
Yes.
because it's so hot.
So I went to graduate school at the University of Arizona in Tucson. And one of my chapters that I never really published on was looking at caves. And one of the really interesting things someone did while I was there was they set up a sonic drip counter so they could hear the drip rate inside the cave. Drip, 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 drip. And what they found is in the summertime, as you say, spring through summer and fall, there's a very clear diurnal cycle. And during the, during the night, the drip rates increased and the water flowed. I guess it's the opposite. It's during the day, the drip rates increased, and during the night, went to the surface. Yep. Same thing. And then hydroponics came up. And so, yeah, I don't know. I've never tried a hydroponic experiment, really. I tried, the closest I've come was picking up all that, like a bunch of those little lesser celandine invasive weeds that are, I think, also poisonous that tend to leaf out this time of year, and just put them in, like, I just used marbles and water to give them some kind of substrate. And yeah, there was sort of like a hydroponic experiment, and they did draw CO2 down for a little while, but then they all just died and rotted. I will say also, when you do this, if you do this and CO2 levels go high and you start to see mold, be sure that you open up the tank outside, because it's horrible. Like, if it goes anoxic, it's really, really, really awful. And so you're in Arizona, Mandy. Yeah. Yeah, so the challenges are going to be different. So for us, it's light. And so we've had to use grow lights and position the tanks carefully. But for you, we've tried some of these in the summer to get a sense of the challenges you might face in a different sunnier climate. Then the challenge is overheating and killing the plants. And they can, these tanks will get up to 125. So, a clever group of students might figure out a cooling system that has coils that maybe come out of the tank and go underground and pipe back in colder air.
Yeah.
Spinach leaves, yes. So just leaves.
A lot of our
gardens
a lot of our gardens, we put water at the bottom instead of watering at the top, because otherwise you have too much evaporation. Yeah. So they're very familiar with under the ground coil type things. So
there's also a question in the chat about tank size.
Yes, well, we've done it with several different tank sizes, and the results are not really that sensitive to tank size. One of the future products, I guess, outcomes of this project is to have a little calculator so that you can enter in your tank size, enter in your data, how much did CO2 go down by, over what time scale, and then it will calculate for you how many tanks would you need around the world to offset human anthropogenic emissions. And the ballpark figure is going to be something like a continent the size of Australia covered in tanks, for most cases. But maybe some of you will design better tanks and you'd only need a continent the size of India.
And there are more things in the chat. Spinach leaves and bottle to draw down.
Yes. So yeah. So you can do cuttings will work for a little while if you're not running this for very long, and you use spinach leaves, salad, even like hydroponic lettuce. I would imagine would probably do enough photosynthesis to get a nice result. Algae and mosses have been the most successful consistently. Not all of them, but almost all the algae and mosses have been successful. CO2 detector and a light. Try wrapping foils. Oh yeah, you can shut off the light. You can get a really nice. I think we did this. We did a workshop up in Syracuse. We imposed really short cycles of light and dark. And let's see if I can pull that one up because it was kind of interesting how quickly you see the response. Yeah, here it is. No, this was not the one. Hold on, sorry. The problem with doing too many of these experiments now. Yeah, so this has a nice diurnal cycle to it. This is not the one with the rapid diurnal cycle, but the light is on for 8 hours, starting right at about the time that the daylight was no longer available to the tank. And you see you get this nice, it gets about 4 hours of natural light in the mornings, and then right towards the evenings when the light goes off, I think we had it shut off at like 8 o'clock, yeah, right at 8 o'clock, that's when the CO2 levels start going back up. Like a nice 12-hour cycle in total of CO2 levels going up and down. There was one that we ran for, I think, 3 weeks or more. Oh, I think I just saw the one with the 15-minute cycles because I was blocking it with my Zoom view before. Yeah, this was a really successful one up here. This is one of the most successful ones. So this is at PRI with a grow light next to a window. Started off at around 3,000 right after a different experiment. So we didn't even open up the tank. It was like a failed experiment. CO2 levels went really high. We installed the grow light next to it on a 12-hour clock, and it brought CO2 inside the tank down to 30, or almost below 30, 9.5 ppm, to the limits of detection. So it really cleaned up the CO2, really effectively. And then we did one that I just saw. This is the one I was trying to find before, where we just did like a funky 15 minutes on, 15 minutes off to simulate what would happen if clouds were passing by. And this sawtooth pattern that you see, those are the 15-minute cycles. And then at night it goes up, and then when the sun is shining, it goes back down, and then it goes into the 15-minute and the 30-minute cycles. And that was just to determine if we would get a fast enough response for 15 minutes in a classroom. Would you see anything? And you do. I mean, if it's a successful, very photosynthetically active tank, CO2 levels will go down quick. All right, we go back to the chat and see if there's more questions.
Yeah, there's more in the chat asking about soil alternatives like coconut fiber.
Yeah, I mean, test it out. I don't know. We haven't done everything yet. This is what we're trying to figure out, like what works, and thinking about the generation of students that you all are teaching beyond 2050, when they are my age. We are going to need more effective systems for CO2 removal. And the broader ambitions of this project are to get students, kids, young people to think about these complex, chaotic, coupled, dynamical systems to be able to manage CO2 in the atmosphere in sensible and sustainable ways. And there are mechanical ways that you can remove CO2. There are industrial facilities that have been recently developed and they're starting to be tested. It's not clear how scalable they are. And yet we have plants, algae, mosses doing photosynthesis and removing CO2 for free. So if you think about what kind of world, what kind of world does it look like if we do CO2 removal with industrial facilities? Well, probably just looks like a world with more industrial facilities and warehouses, but a world that uses biology, uses plants, mosses, algae, creative uses of photosynthesis that's already been tested for 2-point-some billion years. It's literally a greener world. And so thinking about that second half of the century and having students engaged in the science and testing some of these ideas now that can then percolate in the same way that, like famously Bill Gates played around with computers and then became an important figure in the technological revolution that we've been experiencing. We hope that by students getting involved at a fairly early age in thinking about the carbon cycle, by the second half of the century, they'll have some pretty inventive solutions that help undo a lot of the CO2 releasing that we have done. Water outgasses when heated, so our control bottle varies to oh yeah, that's totally, yeah, you could just do like, this is a great idea. So the last, so I guess it's Sarah's comment. One of your controls that you could do to really dig into this, if you're gonna do algae, is to ask, okay, what if I just had water with no algae? How much would CO2 fluctuate based on CO2 outgassing and absorption, based on temperature? So like you think of it as like opening the Coca-Cola bottle and letting it sit, equilibrate at room temperature, and CO2 is gonna outgas. And that would be a great, you could have that as the control in one tank, or you could do that ahead of time for a week or two to get the baseline variability from the water, and then put the exact same amount of water with algae into it and look at the real photosynthetic effects. It'd be awesome. But these are all great ideas. I mean, this is why we're turning it over to this community science approach, because we want to have this out there and learn from all of you all and your students.
So, Toby, a good segue then is Holly asks, do you have programs through which students could reach out virtually to thought partner as they develop their experiments, which sounds like, I don't think we have something in place, but sounds like an aspiration.
I mean, I think this is a great idea, and we are working on some funding mechanisms that would get graduate students. We have one graduate student who's an early participant, who's, I don't know if is Frank here actually? No, I didn't see Frank's name. We have one graduate student who's on board with this. The idea would be to have more graduate students and a larger team of people who've done enough of this to be able to, I guess, thought partner or make a mentorship model, or a coordinator and someone who can answer questions. And I plan to, at the end of either at spring break, but more likely at the end of the semester, I'm going to set up like a Discord channel for anyone interested. Discord server, I guess it's called, as I've learned from my children. So it's a closed discussion board about this kind of stuff. And you can, so if you go to this page, if you go through this, and anyone can sign up for this, and just let me know and I can add you as an educator. But if you go to the homepage here, there already is a bit of a discussion board that we've been using for some of these experiments. And the other cool thing about this portal, the company that we've been working with has added the ability to put some meteorological data in there as well. So if you happen to have a weather station near your school, you can compare the sensors and what's happening in the classroom or outside your classroom to what's happening at a nearby weather station. But yeah, here's some discussion from last fall and links to labs that we've done, or that participants in this project have done. I see that my computer's battery is getting very low, so I'm gonna apologize if I disappear. I'm gonna try to find the cord. But please, more questions or discussion is very welcome. I'm still here. I'm just searching for my cord.
Put in the link to the air quality portal. You want to use the you have to be logged in to access the.
All right. There we go. No, we can, yeah. Now I'm plugged back in. The other thing I was going to show you, so we want, I think, again, this is thinking a few steps down the road, but I had a high school student working with me over the summer. As I mentioned, my own kids have been dragged into this at various times since the pandemic. And one of the things that we put together that we'll share later this semester once it's complete is a little bit of a template for writing this up like a scientific paper. And is this the right one? This is not Google. Just show you what that looks like. My pandemic era Zoom skills have gotten rusty, I can tell. I'm not quite as able to efficiently navigate between screen shares and windows. I was trying to draw something earlier today for a prospective student using the whiteboard. I was like, God, how did we do this for 2 years? So, the way that I write papers, my students and my colleagues write papers, we usually start with the figures. And so this is like a demo from just one tank experiment that went on for 2 or 3 weeks. I think it was the one that I showed you, the really successful one that I showed you. So I made a little diagram of the tank and all the components. And then this is the spectrum of the grow light, which I was just able to get from the company's webpage. And then I made it look pretty. And then this is the data that I just showed you. And so I started by writing figure captions here, and then went back working with this undergrad or the high school student, working together to come up with a description, so an outline of the purpose of doing it, the figure captions, data and methods, so that at the end of the day, what we should have is what is very, very realistically or reasonably like the kind of peer review paper that we would work on. And the reason is
to share the link for that.
What's
Toby,
that?
Can you share the link for that?
Yeah, I want to finish it before I share it. It's a little bit too messy right now. I just wanted to show that it's in development. I can share the figures. That's fine. I can put those in the link in 1 second.
I think we had listed this as a 1-hour session for now.
Oh, we are past it now, aren't we? I think I had also advertised it that way to my wife.
You are still on campus.
So those are the figures. And I just did I tried to do it as much like I would if I were a high school student. I just did screenshots of the graphs that I showed.
Thank you, sir. I think maybe we will call it a night.
All right. Excellent. So long, everybody. Thank you. Turn it over. I'll close out a little tank view.
Good night, all. And if you do try it with students, let us know how it goes.
Yeah. Well, we'll have a